Good morning, good afternoon, and good evening, everyone. Thank you for joining us for our December of 2020 Global One Health Initiative webinar, supported by the NIH Fogarty International Center. The topic for today is pharmacology in One Health, and we will be hearing from speakers Dr. Shridhar Narayanan from the Foundation of Neglected Disease Research in India, presenting AMR and One Health, and Ermias Addo, a PhD candidate from The Ohio State University here in the US, presenting Natural Products in Cancer Drug Discovery, study on the bioactive constituents of the Vietnamese medicinal plant Streptocaulon juventas. And our panel discussion moderator will be Dr. Mark Mitton-Fry from here at The Ohio State University. And he is also a GOHi executive committee member. And with that, I will hand it over to you, Dr. Mitton-Fry.
Great. Well, again, good morning, good afternoon, and good evening, everybody. It's my pleasure to be here today. I'm an assistant professor in the Division of Medicinal Chemistry and Pharmacology in the College of Pharmacy here at Ohio State. So it's my pleasure to introduce our first speaker, Ermias Addo. Ermias is a PhD candidate in my division of Medicinal Chemistry and Pharmacology. He received his Bachelor of Pharmacy degree in July 2010 from the School of Pharmacy, the College of Health Sciences at Addis Ababa University in Ethiopia. In 2015, he received his Master's of Science degree from the same university, and that was in pharmacology. During that program, he did a research secondment in the laboratory of one of my colleagues, Dr. Lour in pharmacology. And then in 2016, he joined Ohio State PhD program in pharmaceutical sciences under the supervision of Dr. A. Douglas Kinghorn. Ermias has co-authored 3 research articles, 2 reviews, and 1 book chapter. He's also been the recipient of several awards, including the Raymond Doskotch Graduate Fellowship in Medicinal Chemistry and Pharmacology, and he currently holds the very prestigious Presidential Fellowship from the Ohio State University Graduate School. His research interests are in the isolation and structural identification of natural products from various natural resources, such as traditional medicinal plants, for the treatment of a variety of different diseases, including cancer. And with that, I will turn it over to Ermias, and thank you very much for your presentation today.
Oh, Ermias, I think you're on mute.
Share screen. Can you hear me now?
Yep, and we can see your screen.
All right, okay. Good morning, everyone. Thanks so much for inviting me to give this talk. My name is Ermias Addo. I am a PhD candidate in Dr. Kinghorn's laboratory at the College of Pharmacy. So first, I'd like to thank the coordinators from the Global One Health Initiative for inviting me to give this talk. So today I'll be discussing on the role of natural products in cancer drug discovery, and with that I'll give my presentation on one of the plants that we currently studying. Here are the outlines for my presentation. I will start on cancer, brief overview on hallmarks, epidemiology, current treatment, and caveats. And the next topic I will discuss on natural products and their role in drug discovery, in particular in cancer chemotherapy. The third one is I'll briefly discuss on how or the kind of method we use to isolate the responsible compounds from the plants or the extracts we test on. Then I'll discuss on one example of the plant we studied by using this method. By the way, can you hear me clearly?
Yep, perfectly.
Okay. All right, so let me start on cancer. So Cancer is, as you might be familiar, is not a single disease. Rather, it refers to a group of more than 100 diseases, which are characterized by at least 6 traits, such as resisting cell death, inducing angiogenesis, sustaining proliferative signaling, evading cross suppressors, enabling replicative immortality, and perhaps the most dangerous one is activating invasion and metastasis to other sites or other organs. So in terms of epidemiology, or how widespread is a disease, cancer constitutes or is the second cause of death in the US. So only next to heart diseases, as you see, it has equal prevalence in terms of death in both males and females. So this shows the gravity of the problem we have from cancer. So there are predictions on the new cases for the 2020, the data, and it has been estimated that close to 1.8 million people are going to have a new cancer case. And this pie chart shows the total number and the breakdown of the major cause of or the major cancer types. And those are breast, colon, prostate, lung and bronchus, and skin. Those are the predominant form of cancers in the US. In terms of deaths, estimated deaths for 2020, more than 600,000 deaths are expected to occur, and these are again from the major causes of cancer or the major cancer types, such as breast cancer, colon, prostate, lung, and bronchus, and also skin. So if this is a problem, do we have treatments? Of course there are, and we can grossly classify them or generally classify them into 3. The first one is surgery, radiation is the second one, and chemotherapy. Each one have its own advantages and limitations. For example, surgery is usually preferable before metastasis, and radiation, if it's beneficial, it has some severe side effects. Perhaps the mainstay of cancer treatment is chemotherapy by using selective drugs, and these have several advantages and limitations as well. So this, even though it has several advantages, it also comes with some shortcomings, such as resistance to the drugs that we normally use, and these are resulted to look for another better alternatives. And also perhaps the well-known problems with chemotherapy is severe toxicity with the drugs. So the area of cancer research for new drugs is always active in terms of finding new drugs which are capable of overcoming these 2 shortcomings that we see. So for that purpose, what kind of source do we have? So we can have synthetic chemistry, for example, just synthesizing compounds from scratch, or inspired by other compounds that we have. Or the another option we have is to look from nature, which usually have some sort of compounds for example, for its defense purpose. So this leads to me to the next point of my talk, which is the role of natural products in drug discovery and cancer chemotherapy. So let me start with the general one, a bit wider one, which is natural products in the drug discovery area. So this pie chart shows all small molecule approved drugs from January 1, 1981 up until September 30, 2019. So the legend is shown here. In terms of natural products from this legend are those included in the highlighted area, usually the top ones. So if you add all these together, these are related to the natural product in one way or another. They are either directly the natural products, or they are inspired from the natural products. So if you count all of these, the value of natural in the product discovery is still significant, and natural products still continue giving us new medicines for several diseases. So to give you an example of perspective on this one, for example, on January 10, 2020, this year, the FDA approved cocaine hyaluronic chloride as a nasal solution for local anesthetic. So cocaine, what is cocaine? Cocaine is just an alkaloid which is purified from two varieties of retroxylem species. One of them is shown here, retroxylem novogratans, and these leaves of this plant usually provide this alkaloid or cocaine. So this is one example of the value of natural products in drug discovery, and this is just approved in January. To give you another example, this is directly from the FDA website, and it says FDA approves only drug in US to treat severe malaria, and this drug is called artesunate. And this is used to treat malaria in adult and pediatric patients. So what is artesunate? So artesunate is not directly a natural product by itself, but it's a derivative of natural product. So it is the precursor for this drug is called artemisinin, which was isolated from the annual or sweet wormwood that you see the picture shown here. And one of the scientists who discovered this drug or artemisinin, artemisinin has been the mainstay of treatment for malaria, but resistance developed. So researchers look for alternatives that overcame the problems that artemisinin has, and they were able to arrive at artesunate by a semi-synthetic method. So artemisinin was discovered by Yu Tu from China Academy of Traditional Chinese Medicine, and she shared the 2015 Nobel Prize in Physiology and Medicine for her discovery of artemisinin from this plant. So these two slides showed or give you an example how the natural products continue in providing us drugs to treat some of the diseases that we face. So with this, since I am more interested in the cancer drug discovery area, what is the role of natural products in that area? So this leads me to the next topic, which is natural products in cancer chemotherapy. So this bar graph is from the same reference, and it shows all anti-cancer drugs from 1946, that is the year probably nitrogen boosters has been introduced into the clinics, up until September 30, 2019. And this is based on the classification based on the source, and again the legends of the key are shown here. And those related to the natural products in one way or another are highlighted in red arrow. And again, if you count all of these together, the value of natural products actually increases in terms of percentages. So there are multiple ways natural products can provide drugs for cancer chemotherapy. One is they can be used directly as without modification, that is the N categories that you see here, or they can be a source of inspiration for others, such as in the S star or S star NM that you see in these two categories. So to give you again another example of the role of the continued role of natural products in cancer chemotherapy, let me show you another example from FDA website. So it said the FDA approves, I'm going to use trade name Enhertu here for patients with metastatic breast cancer who received 2 or more prior anti-HER2 based regimens in the metastatic setting. So what is Enhertu? This is from Daiichi Sankyo and AstraZeneca, and this is the first antibody drug conjugate containing a warhead or payload right from the medicinal plant. And this is just a derivative of this compound called camptothecin, isolated from this plant, Camptotheca acuminata. So what is camptothecin? So camptothecin is again nothing but a natural product. More specifically, it is an alkaloid, and this was isolated in 1966 by doctors Merr and Masukiboni from the happy tree or cancer tree, Camptotheca acuminata. So this picture shows both researchers holding a huge solution of, I don't think nowadays it's possible to have this kind of, or this amount of sample or drugs. So anyways, the blue fluorescence that you see is camptothecin under UV light. So, however, even though it was very toxic to cancer cells, there were some problems with camptothecin, such as it was not soluble in aqueous solutions, thereby prohibiting or preventing delivery by aqueous solutions. So the researchers looked for another way to deliver this drug, and they were able to find an alternative drugs, potent drugs. But this drug, as it is, has to stay or set aside for a long time up until they were able to produce a drug that I showed you earlier, or another name is trastuzumab deruxtecan. And as you can see, the one in green is directly a derivative of camptothecin. So what they did is they linked this drug into with an antibody called trastuzumab, and this directly targets HER receptor. So this is our antibody drug conjugates are just one way of delivery system, so that this one reduces side effects. It also overcomes some of the problems with camptothecin. So this shows the value of natural products in both in the general drug discovery area as well as in the cancer chemotherapy. So the lab I'm in, which is Dr. Kingor's lab, has a project called the P1 project, and that project is based on this success of natural products in cancer chemotherapy. So this leads to the next topic, which is a generalized scheme of isolation that we use in our lab, and it's called bioassay-guided fractionation and isolation. It's not the only way, but it's one of the major methods that we use. So this is the P1 project, which is led by Dr. Kingor on my lab, and this is a multi-institutional collaboration between at least 3 universities, such as the Ohio State University, University of Illinois at Chicago, University of North Carolina at Greensboro. And there are several labs in these 2 units. We are currently here now, so which is led by Dr. Kingor, Dr. Karkache, and Dr. Lour. So how do we isolate the compounds responsible for some of the plants that we collect? So the general process that we use is called bioassay-guided fractionation and isolation. It's simple. What that means is, I'll explain it in a few words. The first thing to do is select the biomass, then you collect. It could be from, when I say biomass, it means that it could be plant or microbes or even animals. They are collected and processed accordingly, such as dried, and these are extracted, and that will lead to an extract. The question that is that how to proceed further, right? To whether this plant or extract have an activity or not, and that will be done by testing for the appropriate biological systems. And the question is, are they bioactive or not? If no, then we will stop here. If yes, we will proceed until the bioactive compounds responsible for the activity of the extracts are isolated. So the next step, for example, is successive partition with organic solvents. Then again, these are tested. Let's assume that the chloroform partition is the most active one. then we'll do the same process or fractionate using several chromatographic methods until we have the pure bioactive compounds, which are characterized by several spectroscopic methods such as NMR. It is after that we test the bioactive compounds we isolated, so that we answer whether these are the responsible compounds for the bioactivity of the extract that we saw. So with this, let me give you the use of this method I explained in terms of in studying one of the plants that we studied, and it's called Streptocaulon juventas, and this was collected from Vietnam. So in terms of the botany, ethnobotany, and chemistry of this plant, this is just a climber or liner which is distributed in some provinces of China and also in Indochina, such as Laos, Vietnam, and several other places. It has been used for traditional purposes of such as anemia, chronic malaria, rheumatism, and several others. The chemistry is also somehow studied, and this plant is known to contain a group of compounds such, called cardenolides. I will come on this one just later on. So this plant, for the current study that we did, it was collected from Vietnam. Vietnam here. There are one of the places called the Nha Trang National Park. It was collected by Dr. Soyarto from University of Illinois at Chicago. It was collected, dried, and it was imported to US. So once collected and appropriately processed, the stems were extracted with methanol, and the extract was tested on one of the human colon cancer cell lines that we use. And we got an IC50 of 2.5 microgram per ml. So our cutoff is usually 20 microgram per ml. So based on that, this plant was found to be very interesting to proceed on. And then for further isolation purpose, we use the same method, the method I explained earlier, and we added additional cancer cell lines to make our study rigorous. So including melanoma, ovarian, and breast cancer cell lines. So based on this method, we were able to isolate several cardenolides, and we studied the preliminary activity relationship of these compounds based on the sugars and some of the chemical features these compounds contain. We are also able to study the in vivo activity of these compounds, and I'll come later on that. So before I proceed, let me explain what cardenolides are. So cardenolides are nothing but just one example of natural products which are simply based on a steroid skeleton. So this usually have three things in their structure: a steroid skeleton that you see here, a sugar at position 3, and a lactone. Because of this lactone, these compounds are usually classified into two. One group contains a 5-member unsaturated lactone, and these are called cardenolides. And the best example is digoxin from the common foxglove that is used for treatment of heart failure and atrial fibrillation. Another group is based on a 6-member unsaturated lactone. These are called bufadienolides, and an example is bufalin from this toad. They are called bufadienolides because they were isolated from this toad species. So these are just like a brief overview of what cardenolides are. So from our current study, when I come back, we were able to isolate several compounds related to cardenolides. These are, like previously I mentioned, because they contain 3 features: a steroid skeleton, a lactone, and a sugar here. We can classify them as those without any sugar, as those with 1 sugar as the position 3, 2 sugars, and 3 sugars, or even those with different kind of connectivity of the sugars. I'll come later on what this 17 beta lactone means. Another group we were isolated. So In terms of the orientation of this lactone, again, the cardiac glycoside can be classified into two, those with 17 beta lactone and those with 17 alpha lactone. What this means that when you draw the 3D structure of this steroid nucleus, the unsaturated lactone can either point above the plane of the steroid nucleus that you see here or below. So from this group, we were, from the second group, that is from the 17 alpha lactone group, we were able to isolate those with a sugar, with one sugar, and with two sugars. So once we have characterized these compounds, we tested these compounds for against the 4 cancer cell lines I mentioned earlier. These are the melanoma, ovarian, colon, and breast cancer cell lines. And this bar graph shows IC50 plotted against the compounds. And for positive control, we used the non-drug paclitaxel, as this is very toxic to cancer cell lines. So from this, the main conclusions from this paragraph are 3. One is that those with sugar are more toxic than the other ones. The best example is K1, compound K1 versus the K3, K5, and the remaining ones. So that means the sugar is important for activity. The other main conclusion from this paragraph is that if you see some of them compounds are not active at here, so that means that the orientation of the lactones at the 17 carbon is very important for activity, because those with 17 alpha lactone are completely devoid of any activity. The other one is, if you notice that these compounds have differential activity against the cancer cell lines based on the sugar type, number, and connection sequence. That means if you want to produce a cardiac glycoside with selectivity, then you can manipulate that by varying the type and number of sugars. So this is their activity towards cancer cell lines. As I explained earlier, one of the problems with cancer drugs, anti-cancer drugs, is their toxicity towards normal cells. So we tested some of the compounds against a normal breast cancer cell line, and this bar graph shows the IC50 or the selectivity index of the compounds against the actual compounds. So the selectivity index is calculated by dividing the IC50 for normal cells by IC50 for cancer cells. So if I give an example, with this larger green bar graph, you see compound K12 is approximately 90 times more selective to cancer cells than normal cells. So this is to mean that if you generalize to mean less side effects, that means if the compound is more toxic to cancer cells than normal cells, that means we can reduce the toxicity, which is really important if you want to proceed into clinical trials that assess reduces side effects. So if you have to select compounds from this to proceed on, the least compound will be probably this one, because it's not that active and it doesn't have that much selectivity index. But the most one you might want to select is probably K13, because it's not toxic to the normal cancer cell lines, but it is toxic to cancer cell lines. So this is a kind of sometimes becomes subjective, but it really depends on the project that you have. But if you want to proceed, this might be a good option to proceed to the next step. With this, we are also, this is the previous slide I showed you on the in vitro cell lines. How about in vivo activity? So we tested the in vivo activity of these compounds against zebrafish embryos as in vivo model. Why zebrafish? As you see here, it has several advantages. They are amenable to high throughput screening because of their small size. High fecundity gives a powerful statistical results, and also some of them other advantage includes their genome is usually 70% similar to humans. So in our study, we looked for two endpoints. One is delayed hatching of the zebrafish embryos or any developmental defects. That by developmental defects means any problems, for example, like this kind of curved shape as opposed to this normal shape. This slide shows the activity or the pictures of the cardiac glycosides tested against zebrafish embryos at 50 micromolar, and we have two controls, negative control and positive control. So as you see for the negative one, the embryos are kind of straight and doesn't have any developmental defect. However, if you see the positive control cyclohexamide, they were unable to hatch, the eggs at least are unable to hatch the 24 hours. An example of developmental toxicity is shown for compound K5, which has this curved developmental defects, which is called lordosis. So of all the compounds that we tested, only 3 compounds were not found to be toxic, as you can see here. Since I cannot show you the structures on this because it will be congested, I have produced another slide with their structure. So what you see in red are toxic to zebrafish embryos, either by delaying the hatching or by causing developmental defects. Those in blue are not toxic. So as you can see how a single difference of structure causes a dramatic change of activity. An example is for K6 and K5, where the only difference is the presence of this methyl group. And this dramatically changed activity from not toxic to toxic in the case of K5. So we were intrigued why these two were show this kind of differential activity. So we did several mechanistic studies. For the sake of time, I'm not going to discuss that time, but the results of this has been published on Journal of Natural Products. So this concludes part of my talk. Sorry, I think I took time, but before I wind up my talk, let me summarize what we found so far. So from the current study for the second part, which is our study on the stems of Streptocaulon juventas, we can conclude that cardiac glycosides are the main compounds responsible for the activity of the extract that we obtain from this plant. And based on that, what I showed you, the structure activity relationships of these compounds has been established. And if one wants to proceed with these drugs, they can do so by varying the number, type, identity of the sugars, even their connection. And also we have seen that the configuration at the C7 is important for activity. From the in vivo model, we can conclude that in vivo toxicity might be reduced by appropriate selection of the sugars. So with this, I would like to thank I would like to finish my talk. There are several people and institutions that were involved in my research, and I would like to thank all of them. First is my advisor, Dr. Kingon, my committee advisors, Dr. Kingon, Dr. Karkache, Dr. Fuchs, Dr. Liva, Dr. Ili from Dr. Kingon's lab for the guidance, my former students, as you see, my deepest gratitude also goes to Dr. Craig McCroy for the help in the instrumentation, Dr. Karkache for doing the in vivo, some of the in vivo zebrafish toxicity assays. And also perhaps my gratitude goes to Dr. Soyarto from UIC for, and also Dr. Trump from VAST from Vietnam for plant collection and identification so that we continue. Dr. Jana from is also thanked for her for doing some of the cytotoxicity studies. And I would also like to thank the P1 grant from NCI NIH, Presidential Fellowship from the OSU Graduate School, the Raymond Doskovich Fellowship from Division of Medicinal Chemistry and Pharmacology, NGT appointment from College of Pharmacy. With this, I finish my talk for today. Thanks so much for listening. I'll be happy to take any questions if you have. Thank you.
Thank you so much, Ermias, for a really wonderful talk. We'll do questions during the panel discussion after the second talk.
All right, great.
So if you could stop sharing then. Great, thank you. It's now my pleasure to welcome our second speaker, Dr. Shridhar Narayanan. He is the Chief Executive Officer for the Foundation for Neglected Disease Research in India. He has over a decade of experience in drug discovery and development expertise in the Indian pharmaceutical sector in a variety of therapeutic areas. He holds a basic degree in pharmaceutical sciences as well as a PhD in pharmacology from the Ohio State University. Also has postdoctoral experience from the University of California, Los Angeles. He has served in a variety of executive level positions in the pharmaceutical industry, and especially has led the discovery and development of 2 potential clinical candidates for tuberculosis and malaria. Throughout his career, he's contributed to the discovery and development of 15 clinical candidates. For those of you who are not involved in drug discovery, that is an absolutely outstanding track record of productivity. He also serves as a scientific consultant and advisor to biotech companies and research institutes, and holds an adjunct faculty position at the Transdisciplinary University in Bangalore. So with that, I will welcome Dr. Narayanan and invite him to share his screen and begin his presentation.
Thank you very much, Mark, for that kind introduction. And it's good to be back at Ohio State, even though it's just virtual. So my topic today is going to be on AMR and One Health. Before I get into the meat of the topic, I just wanted to talk a little bit about my organization and what we do. Is my screen visible?
Yes.
Yeah, yes, it is.
Wonderful. So let me, all right. So my organization is a not-for-profit company, and we are exclusively devoted to discovering new drugs, diagnostics, and devices, which would help in the treatment or detection of a variety of infectious diseases. And we focus a large part of our work on the drug-resistant pathogens. This is our R&D center, which is located in Bangalore, India. It's about 10,000 square feet of lab space with facilities for both biosafety level 2 and biosafety level 3 organisms, which include bacteria, fungi, viruses of all kinds. And our most recent feather in the cap is that we are probably one of the few organizations in India which are capable of doing the SARS-CoV-2 research. So, we have access to the SARS-CoV-2 virus, including the in vitro assays and the animal models. I have a total of about 20 scientists and support staff who work with me in this facility. And in addition to all the microbiology related work, we are able to do the drug metabolism and pharmacokinetics, all the bioanalytical work, and even the medicinal chemistry. We have our own labs where we do some of the synthesis of the compounds that I'm going to talk about today. So in terms of the pipeline of molecules that we have, we work mainly on diseases like tuberculosis, malaria, dengue, and leishmania. And as you can see, we have a broad pipeline in terms of different stages of clinical development. And our candidate, which is the farthest in development, is currently in phase 2 clinical trials in humans. And this is a molecule for the treatment of tuberculosis and is being developed in partnership with TB Alliance and the Gates Medical Research Institute. These trials are currently ongoing in South Africa. In addition to this candidate in phase 2, we have a few other clinical candidates which are being repurposed. One is a cancer compound called saracatinib, which we are repositioning for tuberculosis. And the second one is a cancer compound, is an anti-malarial compound, chloroquine, which is also being repurposed for tuberculosis. In addition to that, we have a variety of other programs which are from various natural and synthetic sources of chemistry, which are in development for these diseases. In terms of our diagnostics pipeline, we have one diagnostic which is for tuberculosis, which recently we have completed a 250 patient clinical trial. And all of our diagnostics are meant to be diagnostics which could be applied point of care in a primary healthcare center in a rural setting, not only in India, but any of the developing nations. We also have one diagnostic used for detection between bacterial versus viral infections. And I'll talk a little bit about this in the later part of my talk. We have a special interest in gestational diabetes, and we are looking at developing a blood-based diagnostic which could potentially predict the risk of gestational diabetes even as early as the first trimester. And I'll also talk about the device that we have for antibiotic depletion. So when I was talking to Laura about the scope of this webinar, and she mentioned it's pharmacology and One Health, so I thought it's probably interesting to talk about the different aspects of One Health and how my organization is contributing to that. So in terms of our efforts, today we are actively involved in discovering and developing drugs for a variety of human diseases. And we also are working on devices which will ultimately help the environment. And we've just started a program on mastitis with one of our molecules, which is looking at animal health. So as an organization, we definitely want to contribute to the One Health concept and want to make sure that whatever we do falls in line with the intersection of all of these three areas. Mark mentioned about the drug discovery process, being a long one. And I just want to give those of you who probably are not from this area a little bit of flavor of what it does take and the timeline that it takes for actually discovering a new drug. Most of you probably have heard about the SARS-CoV-2 vaccine story and how the development of the vaccine has happened within a year. I think that is a rare event. Most drug discovery efforts take anywhere between a good 8 to 10 years. And right from a target identification, lead generation, lead optimization, and all the way to completion of phase 3 and launch in the market, it can take anywhere up to 10 years, right? So my organization works primarily in all the areas right up to preclinical development. This is all work that is done in-house. And for the rest of the phase 1, phase 2, phase 3, we outsource the work or work with partners who are capable of doing this. Now, the reason why we want to do this is because this is where most of our expertise is, and it's easier to do this at a much efficient manner in a lower cost, right? The kind of dollars that are required to develop a drug, whether it is for oncology or whether it is for a bacterial infection, is anywhere in the range of $800 million to $1 billion. So with that kind of money being required, I think it's very difficult for an organization like mine to take it all the way to the market. But what we definitely want to do is do right up to phase 1 and then find a partner who will be able to take it forward into clinical development. So the first project that I want to talk to you about is a small molecule broad spectrum antibiotic that we have discovered in-house. Now, why do you want to look at a broad spectrum antibiotic and what is the role of microbial resistance in this? So before we go into that, I just want to give you a flavor of how a resistant bacteria develops, right? So when you have a non-resistant, initially it's a non-resistant bacteria, they're multiplying by millions. There are some mutations which happen in the DNA, which is a natural process. And these mutations make the bacterium drug resistant. And in the presence of drugs, only the drug resistant bacteria survive. Now this population then multiplies and thrives. Now, the other way in which the drug resistance can happen is where the drug resistance bacteria actually transfers the gene to a normal bacteria. And when these bacteria multiply, you're having a total drug resistant population that is formed. Now, why do we want to address this problem? A, there is a fast emerging antimicrobial resistance, particularly the growing threat of carbapenem resistant E. coli and other drug resistant bacteria like Acinetobacter and Pseudomonas, which are highly prevalent in the developing world and also in the developed nations. There is a huge economic burden that the AMR is bringing to the table, and it's almost to the extent of $34 billion in the US alone. In Europe, it's estimated that they would be spending close to $105 billion per year for the antimicrobial resistance. So there's an urgent need to control the antimicrobial resistance and to reduce the number of infections because of antimicrobial resistance. Now, the target product profile that we have when we started this program was to target one of the organisms of the critical priority list of the WHO or the CDC. In terms of the chemistry, it had to be a novel patentable series with the potential for structure-activity relationship. In terms of its microbiological profile, we wanted it to be active against the Enterobacteriaceae, especially the E. coli, Klebsiella, and Proteus, including ESBL producers, with no cross-resistance and a low mutation frequency. Now, this was essential for the molecule to have a longer life in the clinic. In terms of the drug metabolism and pharmacokinetic parameters, we wanted to have a pharmacokinetic data which would support its use in all systemic infections. And in terms of safety and tolerability, we wanted it to be comparable to the current cephalosporins, which are probably one of the safest class of antibiotics. So what we've managed to do is develop a first-in-class small molecule, which is novel and patentable and targets the WHO critical priority pathogens with desirable drug metabolism and pharmacokinetic properties. So just to give you a feel of the data, so this is data from the priority 1 critical pathogens from the WHO priority pathogens list, which include Acinetobacter, Pseudomonas, and Enterobacteriaceae. So we have data here from the Pseudomonas and the E. coli and Klebsiella pneumonia. Now you can see that our molecule has an MIC of 0.37 microgram per ml against Pseudomonas aeruginosa. Very, very potent against the E. coli, which has both KPC positive and NDM positive. So this is probably one of the most resistant bugs that you will find. Also against a different strain of E. coli, again with the same profile of 0.09 microgram per ml, and also Klebsiella pneumoniae at 0.37 microgram per ml. So it's very potent against not only Pseudomonas, E. coli, and Klebsiella, and at a very, very low concentration. In terms of its NDM profile, so this is the proof that it is both NDM positive and KPC positive. So you have values fold which are 8 to 24-fold higher in E. coli and more than 80-fold higher in the Klebsiella pneumoniae. So which indicates very clearly that it has NDM positive and a KPC positive profile. Now, we also looked at a variety of other strains which were carbapenem sensitive. And in all of these strains, except for 2 of them, we had a very potent MIC against Klebsiella, Acinetobacter, Pseudomonas, Enterobacter, Salmonella, and even E. coli. And this is the resistance profile of these strains of organisms, where you had some which were sensitive to all the drugs, resistant to just a couple of them, and some which are resistant to a whole bunch of them. Now, in terms of organisms where we didn't know the carbapenem status, we had a variety of organisms which belong to Pseudomonas, Salmonella typhi, Proteus, Shigella, E. coli, and against all of these organisms, this molecule was highly active. So we had a series or a molecule which could potentially be developed into a drug-like candidate with a very potent profile against a variety of microbes. Now, we also looked at the gram-positive organisms, and in the gram-positive organisms, it was effective against the Staphylococcus aureus, but not as effective against the Enterococcus species. So in terms of its profile, we would like to improve its profile against the gram-positive as well to make it a broad-spectrum antibiotic, or reduce it to being focused exclusively on the gram-negative organisms. So we also looked at the kill kinetics of this molecule in the E. coli, and you can see the control, this which is the red line, where you can see a very nice growth of the organism over the 6-hour period. And these are various concentrations of our molecule. And you can see at as low as even 0.14 micrograms per ml, it starts to show at least a 1 log kill. And we have reached a maximum of 3.5 log kill at a 1 microgram per ml concentration. In the same species, you have the moxifloxacin, again showing you a response which is an Emax of about 5 microgram, 5 times 5 log kill. So very clearly, this series is a fast killer and is also potential for a wide variety of organisms. So what we are looking at is developing this series for further. We want to make about 50 to 75 molecules in this series, ensure that we maintain the spectrum of activity against a variety of critical pathogens. We want to identify a lead with good DMPK properties, efficacy, and safety properties, and optimize this lead to identify a clinical candidate within the next 12 to 18 months. So having talked about the drug discovery program, I want to shift gears a little bit to talk about our efforts in helping the environment. So what we've done is we have devised made a device using waste materials, which will help deplete antibiotics from hospital effluents. As you all probably know, the hospital effluents contain a variety of antibiotics, and this results in a generation of antibiotic-resistant organisms in the water samples. So what we need is something which can take care of the high antibiotic load in the hospital effluent water, which leads to antimicrobial resistance. And there is no active mechanism focused to decrease this antibiotic load in the wastewater. So we found that as an opportunity, which we could use using our knowledge about antibiotics to actually devise a product which could remove antibiotics in an online fashion. We've used waste material such as charcoal and polymers from different natural products, which are incorporated into this device. And this acts as an adsorbent material, which has the capacity to absorb more than 250 milligrams of antibiotics per gram of the adsorbent. And this is all at the lab scale currently. So the adsorbent mix, which has been formulated, can absorb about 250 milligrams of antibiotic per gram of the adsorbent. We've used a variety of adsorbents from waste material like charcoal from bamboo, coconut shell, areca nut, cashew, pine wood, including fillers like corn cob, paddy husk, even human hair, wheat bran, and some earth-like material with additives like paper, cellulose, silica gel, etc. Now, the reason I'm giving you this wide variety is because this is something which we are in the process of patenting. So I'm not disclosing the exact formulation that we have used, but it's a mixture of a few of these ingredients that has gone into the lab scale prototype. Now, just to give you a picture of how good these are. So this is an initial range of antibiotics that we tested: niacinamide, isoniazid, rifampicin, ethambutol, ciproflox, moxiflox. And we use the adsorbent at different concentrations. And you can see that even at as low as 20 milligrams of the adsorbent, you are beginning to see a very strong profile of being able to absorb these antibiotics completely. Ethambutol was one of the antibiotics that we could not manage to absorb with this mix of adsorbent material. Now, as we went along the process, we are able to come up with a newer mix which was capable of absorbing variety of antibiotics, and we increased the antibiotic spectrum to include cephalosporins, penicillins, streptomycin-like compounds, and a variety of others to kind of see if any of these or all of these could be adsorbed using this class of adsorbent mixtures. And we came out with a final mix now, which is mix 3001, which is able to adsorb almost all of them close to 90% or more. Now, what we've also done is taken this adsorbent mix 3001, and we have looked at the number of elutions that we could make of an antibiotic mix through this. And we are able to show that we can do at least around 17 to 18 elutions of the waste material containing the antibiotics through this device. And it is still able to function with the same efficiency that it could do at elution number 1. So with that, we were very encouraged that we had a device which could be used to deplete antibiotics before it actually went into the wastewater, right? So this is how we envisage our final product, which we would be looking at an effluent treatment plant where we could either put this device as an online before the water gets into the effluent treatment plant, or we could potentially suspend this in a semi-permeable membrane inside the effluent treatment plant to allow for adsorption of the antibiotics before it is released into the municipal wastewater. At this point, we are looking at a variety of optimization trials, including the pH of the antibiotic mix, the interaction temperature, and the time for interaction between the water and the adsorbent material. And what we would ideally like is for this time of interaction to be less than a minute, with the interaction temperature close to room temperature and a pH of around 6 to 7, which will allow us for deploying this in any kind of health setup. Now, the third thing which I want to talk to you about is the diagnostic that we are working on, which is being used to differentiate between bacterial and viral infections. Now, why do we need a diagnostic to differentiate between bacterial and viral infections? The main reason is the misuse of antibiotics, where it's either because of a wrong prescription, where antibiotic is prescribed for a viral disease, an overdose or an underdose of the antibiotic, which means that lack of compliance in terms of the number of doses that need to be taken, people taking over-the-counter prophylactic doses even when there is no infection, and all of these lead to actual antimicrobial resistance, right? Now, most of the times, it is an empirical diagnosis made by the physician. And in many countries like India, where even the pharmacist makes a diagnosis of whether it is a viral or a bacterial infection, and either prescribes an antibiotic without consideration of what that infection is coming from. So what we want to do is come out with a blood-based biomarker, which could be administered at a primary healthcare center, and this can, or a pharmacist shop, and this could potentially help minimize the wrong prescription of antibiotics. So we did a variety of experiments, and I'm just going to give you a snapshot of what we've done. So this is from a macrophage cell line called the THP1 cell line, and this is looking at a biomarker called endoglin, and this is a non-infected macrophage, and these are from the infected macrophages. So you can see that when you infect the macrophages, either with a gram-positive or a gram-negative infection, there's a significant increase in the endoglin release from the macrophages. Now, we've also looked at this in an in vivo animal model, where we have infected the mice with both the gram-positive or the gram-negative bacteria. And this is the non-infected animal. This is the gram-positive infected animal where you can see a significant increase in the endoglin level. And this is the gram-negative infection. Again, you see a much higher increase in the endoglin levels. Now, we've looked at this also in the clinical situation. We have an ongoing clinical trial with about 200 patients, which is being run at different hospitals in India. And this is the fold increase over control in a bacterial infected patient and a viral infected patient. So very clearly, endoglin seems to be a marker which could potentially predict or help us diagnose whether the infection is a bacterial infection or a viral infection. So this would enable us to actually prescribe antibiotics in a much more rational manner. So in addition to all of these efforts on the discovery, diagnostic, and the device side, we also do something called as a community connect program, where we work with school students and college students to create awareness about a variety of diseases, especially the infectious diseases. We also do skill building workshops for graduate and postgraduate students in our biosafety level laboratories and help them understand the drug discovery pathways and how drug discovery is actually done and help them with their career enhancement. And finally, we also have educational visits from a variety of students from the biotechnology space, faculty from different universities who come and visit our center, and we encourage getting into research and development for a variety of diseases, including the microbial diseases. So this is just a snapshot of the school health awareness program. Till date, we have reached about 25 schools, and at a cost of less than $2 per student, we are creating awareness on a variety of health-related issues, especially in the infectious disease space. With that, I want to end my talk. Want to acknowledge Robert Koch, who's the pioneer in terms of the discovery of tuberculosis, and want to say, go Bucks, let's go get the Big Ten championship. Thank you very much for your kind attention.
Thank you very much, Dr. Narayanan, for an excellent presentation. At this point, we'll have a panel discussion between our two speakers. So if you have questions for either of them, you could place them in the chat or the Q&A, and I can ask the questions of the speakers. Perhaps as questions are getting started, I could ask a question. Dr. Narayanan, in talking about your new small molecule broad spectrum antibiotic, the data you showed were really exciting. Very potent activity, especially against the multi-drug resistant gram-negative organisms. You did show a couple of isolates with highly elevated MICs, one in Pseudomonas, I think one in E. coli and one in Klebsiella. Do those isolates give you any insight into the mechanism of action of your compounds, or is it some other mechanism that's conferring reduced susceptibility?
So excellent question, Dr. Mark. I think that is a work in progress right now. So we are looking at those specific isolates and trying to figure out their broader sensitivity profile, looking at variety of classes of antibiotics and to see if that would give us an indication of what the potential mechanism could be. We're also generating resistant mutants at this point of time to see if whole genome sequence of the resistant mutant will actually guide us as to what could be the area of action of these drugs, of these molecules, and whether they could potentially be, either protein synthesis inhibitors or cell wall inhibitors, or any of those other classes in terms of activity. So that's a work in progress.
Thank you. I was taking notes during your presentation, but did I see correctly that the molecular weight of your initial lead was less than 200 grams per mole?
That is absolutely correct. So we were also
that is an amazing small molecule. So
it is really an amazing small molecule. And now we are actively starting a program where we are looking at doing an SAR around this core structure And this is a commonly used code structure in the chemical space. So we are very surprised by the breadth of activity. And when we look at the patentability of this code structure, it seems to be very the opportunity to patent is very high. There has been very limited work done on this code structure. Unfortunately, I'm not revealing what that code structure is today, for obvious reasons, but we are very excited by this low molecular weight compound. And we definitely hope that when we do an SAR, that it retains its activity and that we are able to use that SAR to actually modulate the physicochemical properties and the drug metabolism properties to actually make a drug which would be beneficial in a variety of different indications.
Okay, thank you very much. As I'm waiting for other questions, Ermias, I'll ask you a question as well. You described two different strategies for doing preliminary safety evaluations of your natural products. The first involved using a selectivity index where you measured cytotoxicity for a normal cell and compared it with the cytotoxicity in your cancer cell lines. And the second was an in vivo model where you used the zebrafish to look at toxicity. My first question with that is, how good is the correlation that you observe between those two types of assays? Do you generally observe that one compound is toxic to both the normal cell lines and the zebrafish, or do you see divergence of toxicity?
That's a great question, Dr. Mark. I believe the second part you said is correct. So sometimes you will be able to proceed, whatever you find from the in vitro, then there will be a good correlation in the in vivo as well. And you can proceed into further, for example, into clinical trials. However, there are some cases where those two doesn't match. I believe one of the compounds we have is silvestrol. It has good activity. If you know silvestrol, that's good in vitro activity, and I think also good selectivity, but it was toxic to, I believe, dogs. So it was kind of dropped, I think, from further studies, but that's one way. So there will be definitely a disconnection between the in vivo study, the in vitro and the in vivo. Yeah, it can go both ways, but mostly they will have good correlation, but sometimes the disconnection between the in vitro and the in vivo results will definitely stop your research into, for example, going forward.
Okay. I don't see other questions in the chat yet, so I'll continue asking questions while we wait. Maybe a follow-up to that, Ermias. I don't think you showed compound K12 in the zebrafish, or I might have missed it. I did notice that K13 was very non-toxic in the normal cell line as well as non-toxic in the zebrafish model, so that was exciting. But I'm curious about compound K12.
Yeah, unfortunately, we did not test K12. I apologize. Yeah, so I didn't present. Wait, sorry. Yeah, we did not test K12. Okay. So K12 is another compound actually. So it's not active, it's not cytotoxic, so it doesn't have any activity. So that's why we left it out.
Okay. Thank you.
Sorry, Dr. Mark. I confused this with another compound. So K12, it is very toxic, cytotoxic. It has a good cytotoxic activity, but we did not test it to in vivo studies.
Okay.
Sorry.
Laura, do you see questions in the chat yet, or should I continue asking questions?
It looks like you can continue asking questions.
Okay.
Yeah, thank you.
So back to Dr. Narayanan, you showed some interesting kill kinetics for your small molecule antimicrobial compound. And I think you showed the kill curves out to about 6 hours. For some compounds, Ermias, could you mute again, potentially? For some compounds, one sees a rebound in the kill kinetics assay, somewhere between 8 and 24 hours. And I'm curious if you observe that with this compound, or if the killing at 6 hours persists out to a later time point.
So Mark, the kill persists out to 24 hours. We've not seen any regrowth that happens after 6 hours. Our initial kill kinetics experiments were done only to see if it were a fast killer or a slow killer. And we've established that it does kill rapidly, and it stays for 24 hours. So we're not worried about a resurgence or a development of rapid resistance with this class of molecules.
Excellent. So maybe one additional question for me then on the new small molecule antibacterial compound. In the United States, it has recently been very difficult to achieve commercial success with new compounds for multi-drug resistant gram-negative infections. And there are a number of reasons for that. But several small companies recently who have launched really potent, really exciting new therapies have not gone on to commercial success and have gone bankrupt, unfortunately. I'm curious if the same market pressures exist in India, or if the situation is somewhat different there.
Excellent question, right? The same market pressures exist in India as well, right? So it's going to be a difficult uphill task to get it to the market. And most of these companies have gone bankrupt after they've got the approval. So the development part was not the challenge. It was the post-development maintenance of the revenues, which was a big challenge. And if you look at the antibiotic space, it will probably require a good 5 to 7 years before you start hitting peak sales, right? And under those conditions, the initial investment of $800 million, and especially in the antibiotic space, what is seen is that if you have the best molecule, it will most likely be the last line of use, right? It will never be a first line use. So it is only when all the other avenues have completely been exhausted that a physician would prescribe your molecule if it's really best in class. Unlike in cancer, where if you have the best molecule, you probably will become the first drug of choice, right? And so that is probably the reason why there is a lot of attrition in the antibiotic space, especially with molecules which have gone into development, which have been approved by the FDA and are struggling to make it to the initial, I would say, the new valley of death between the year 1 of launch and year 5 of launch, right? So what we are expecting, and especially if you do the drug development in countries like India, where there is a really good infrastructure in terms of hospitals, in terms of availability of patients, and being a chemistry hub, and formulation hub, you can actually manufacture and formulate very easily in India. I would think that you could bring down the cost of the clinical trials significantly. And if you can do that, you would definitely be able to get to the market at a much lower cost than what is seen globally. And I believe that would give you an advantage when you get into the actual marketing phase, where your initial investment has been much smaller as compared to that in the Western world. I hope that answered your question.
Yes, thank you very much. So we do now have a question for you in the chat regarding your detection methods for bacterial versus viral infections from Debasu Gela, and I'm sorry if I mispronounced your name, but how sensitive is ENG as a biomarker to differentiate bacterial infection from non-bacterial or viral infections? And have you compared it with complete blood cell count, or CBC?
Yes, so very good question, and definitely it is sensitive. So we are looking at very clear differentiation in terms of the concentrations of endoglin in bacterial versus viral. Now, this is only one of the biomarkers that I have showed you today. What we have are a couple of other viral specific biomarkers, which we will be able to add to the repertoire to help distinguish between the bacterial and viral, right? So it will be a combination of biomarkers that would actually get used. In terms of comparing it with the CBC essentially will not tell you whether it's a bacterial or viral infection. So if you look at a CRP or a procalcitonin, they are more indicative of an infection and not whether it's a bacterial or a viral infection, right? So even at that point, it is important for somebody who comes in as a patient first time into the doctor's clinic with a fever to be able to be diagnosed with either a bacterial or a viral infection for that prescription to be accurate, right? So I would think that you would need more than a CBC to actually get to that prescription level diagnosis.
All right, thank you. I have a follow-up question on the diagnostic. You shared some interesting data from both an in vivo model and from some human data. It wasn't clear to me though what biosample you were using for your analysis.
So the biosample is just blood. So you could do blood or you could do plasma, right? So the idea is for this to become a point of care, especially for early respiratory infections or any other systemic infection, to say whether it is bacterial or viral. We've not tried with any of the other sputum or other body fluids at this point of time.
Do you envision that it would be something like a finger prick blood sample? It's really straightforward and easy to do?
That is what we envisage this as. So we would look at a pinprick and something like a glucometer-like thing where you just put a drop of blood and you get a readout, and that number or the color, it is dumped. We want to dump it down to the extent where anybody who's even very a not so highly educated person or a technically sound person who can interpret that data and understand whether it is a bacterial infection or a viral infection. Okay. Ultimate thing, it would become a home test.
Excellent. Okay, I'll ask another question of Ermias at this point. Ermias, you've spent a great deal of time in the field of pharmacognosy now, including your exciting results today on these anti-cancer compounds isolated from the Vietnamese plant that you spoke of. What do you consider the biggest opportunities in your field right now? Where's the greatest impact likely to come, say, in the next 10 years?
That's a great question and also kind of difficult to predict. I believe, natural products have a great potential, always have a great potential, and will always definitely give us an opportunity to find new alternatives for the current problems we face, even the antimicrobial resistance, even the cancer area. So with the development of new analytical methods, I believe we may be able to definitely find a new alternative to the existing ones. So if I think the application of these new analytical methods or new way of, for example, molecular networking, combining those kind of analytical methods and with the bioassay techniques, that will definitely lead us to finding new drugs for the problems that we may face. So I've shown you that the case of camptothecin, where it was set aside as a drug by itself, even though it led to some of the potent drugs that we know, such as the Aglycone drugs, such as toxicity and others. Camptothecin wasn't used as it is. It has to set aside for close to what, probably for decades up until it became a drug again, right? So the reason it's where the delivery system was changed so that we kind of avoided toxicity from the drug by itself and also the solubility issue that we faced. So with that kind of development in the delivery methods as well as in the analytical methods, I believe we'll have something in the future. And the good thing in that perspective is identifying the right sources so that whatever we find leads to us a good candidate as a drug.
Thank you. So one follow-up question. My area of research is new antibacterial therapies. You've been working primarily with new anti-cancer therapies. You go through this very painstaking process of isolating bioactive compounds using this bioactivity-guided fractionation procedure. And I'm curious how we can better study your compounds as to whether they might have other interesting bioactivities, whether it's antibacterial activity or activity in hypertension or other disease areas. How do we capitalize on the new molecules that you've identified to explore their scope of therapy more broadly?
Yeah, I believe that that's a great question. So that would be a good question for collaboration purpose, right? So I think that's one of the ways that this kind of actually the big drugs has been discovered. For example, the plant material that was collected in the 1950s, the NCI or USD, US Department of Agriculture, for example, collected thousands of plants for other purposes, such as finding steroids as a precursor for some steroidal compounds. But that means if the researchers during those times, that is Dr. Wall and Woni, didn't look for anti-cancer activity, we wouldn't have computation as a clinical derivative, right? But there was good collaboration, good observation from the researchers for testing the plant extract against cytotoxic activity. So that leads to some of the best drugs that we know so far, such as Taxol, Cont'd, and several other drugs. So as to your question, the good way is to have a good collaboration. For example, if when we isolate some compounds, then I can, besides the primary target we do, that is for cytotoxicity, we can even send you the compounds, right? I'm sure you don't need that amount of compounds that you have. So one way to do is collaboration, so that whenever we isolate compounds, we can send it to you so that it is for others. And this is beneficial in terms of drug repurpose as well. For example, cardiac glycosides have a good track record of clinical data. That means if you want to use them, for example, for cancer or even for other purpose, they have a good pharmacokinetic and pharmacodynamic properties. That means a huge chunk of preliminary data has been already stored, so that the next step is how to deliver the drugs to the patient. So a collaboration would be a great start for your question.
All right, thank you very much. So perhaps one last question for Dr. Narayanan. Regarding your compound for or your absorbent mixture for removing antibiotics from hospital effluent, you showed some exciting data there as well. I'm curious though, what happens with the absorbent material after it's been through its numerous cycles of removing antibiotics, and how you then dispose of that in a cost-effective way?
So excellent question, Mark. I think the reason why we collected all the charcoal and the waste material as the possible adsorbent was that it could then be incinerated without a great deal of damage to the environment. So when we adsorb all of these antibiotics, the device itself can then be incinerated and completely destroyed. So along with the antibiotics.
Excellent, thank you. I don't see other questions. Laura, would you like to wrap things up for today?
Yes, thank you so much, both Dr. Narayanan and Ermias for your wonderful presentations, and Dr. Mitton-Fry for your wonderful moderation and questions. We will, with that, go ahead and close, but we look forward to seeing you all in 2021. We will be sending out our 2021 schedule shortly and posting to our webpage. And thank you. I hope you all have a wonderful holiday, and we'll see you next year. Thank you.
Bye, everybody.
Okay,
thank you
so much.
Bye-bye. Thank you. Thank you for the opportunity. Bye-bye.
Thank you.